Welcome to this episode of Disease Du Jour on the topic of equine muscle disorders. The Disease Du Jour podcast is to you in 2020 by Merck Animal Health. Our guest for this episode is Stephanie Valberg, DVM, PhD, a diplomate of ACVIM and ACVSMR. She is the Mary Ann McPhail Dressage Chair in Equine Sports Medicine and a professor of large animal clinical sciences at Michigan State University. Thank you, Dr. Valberg, for joining us today on Disease Du Jour to talk about equine muscle disorders from a veterinarian's viewpoint.
My pleasure.
Well, let's just start in with what are the most common equine neuromuscular disorders that veterinarians should understand?
Well, I think the one that probably everybody deals with most commonly is tying up or potentially exertional rhabdomyolysis. And I think the thing I'd like to talk about a little bit is a diagnostic approach to those muscle diseases. The thing that has we have always relied on, going back to the 1960s and 70s, the research showed that you could take blood samples from horses and determine whether or not they had muscle damage based on muscle enzymes. So enzymes like creatine kinase or CK and aspartate transaminase that leak out of damaged muscle cells into the bloodstream. And it's really, really useful to utilize those tests in order to try to characterize muscle diseases. And the reason for that is that many horses, owners will present them as historically having had muscle, what they think is tying up or pain and cramping after exercise. But if you haven't got documentation that there was actually muscle damage during those time periods, there could be horses that are having muscle cramping and muscle fasciculations, as opposed to horses that are actually tying up or have exertional rhabdomyolysis with. And the way to be able to do that, obviously, is during the period of time after the horse has had an episode. So usually the best time is somewhere around 4 to 6 hours, or it can be later, to take a blood sample and determine whether or not you've got elevations of CK and AST. Because if that's happening and you document that, then you clearly know that the diseases that you want to look at are diseases that are causing horses to have exertional rhabdomyolysis, as opposed to taking the sample when the owner is reporting the horse is having clinical episodes, and then you find that there's never an elevation in CK and AST. It gives you a different group of muscle disorders that you think that you potentially want to investigate. So I kind of try to then, based on the clinical signs, if owners are reporting horses that have exercise intolerance and they're not going forward and they think they've got muscle pain, try to first categorize those exertional things as, is this an exertional myopathy that's not characterized by elevations in CK, or are we really dealing with exertional rhabdomyolysis that I've been able to document? The other thing that's really important about CKs and ASTs is knowing when you took the sample relative to when the horse had the clinical signs. Because if you take it immediately, you might get some elevations in CK that don't necessarily reflect the amount of muscle damage, because it's going to take 4 to 6 hours before CK will reach its peak values. And then it usually starts to come down. So if you're taking the sample 12 hours or 24 hours later, you also are oftentimes underestimating maybe the severity of the elevation in CK. So I think in doing that, got elevations in CK and AST, you know you're dealing with exertional rhabdomyolysis, then you can start looking at the potential differential diagnoses. So in some cases, it's just an environmental or training issue. We call those sporadic cases of exertional rhabdomyolysis. There isn't any underlying, genetic or inherent susceptibility that the horse has. Something has just kind of happened in the environment, affected the horse's muscles. It's got muscle pain and some damage, and it needs some rest and the ability to kind of get over that. And normally for horses like that, we'll give them some time off, put them out in a pasture so they can move around as much as they want to, rather than keep them confined to a stall as soon as they're comfortable to move around, and then gradually put them back into exercise. And, during that interim time, trying to investigate, were there any training issues? For example, the horse just wasn't fit enough to do the amount of exercise that they were asking it to do. Do we have a good balanced diet with adequate amounts of electrolytes and vitamins and minerals for that horse? Are we feeding excessive amounts of carbohydrate, or is there a good balance of carbohydrate in the diet? And many times after a period of rest and gradual transition back, those horses with sporadic problems will just be fine. Then there are some horses that you do that and you put them back into exercise and they start doing it again and again and again. And in those cases, now we're starting to think this is a chronic form of exertional rhabdomyolysis. What could the underlying basis for this be? An approach to often dependent on what breed of horse you're dealing with, because we now have some nice genetic tests that are a nice pain-free way to diagnose the underlying basis for exertional rhabdomyolysis. And if they are quarter horse related breeds, draft horse breeds, particularly ones that are derived from the continental European breeds, some of the warm bloods. Then we have things like polysaccharide storage myopathy. We can do a genetic test for type 1 PSSM and get potentially an answer to why the horse is tying up. In quarter horses, we could do a genetic test for malignant hyperthermia and get it potentially an answer for why horses are tying up. And then we can take the appropriate steps to modify the horse's diet, knowing that the horse has PSSM1. If those genetic tests are negative, then, and the horse keeps having issues with tying up, then we're often thinking about whether the horse has another recurrent cause of exertional rhabdomyolysis. And when I'm looking at those horses, I like to try to look at their history and their temperament and their breed and figure out what is most likely going on. So with race horses or endurance horses or warm bloods that have this sort of nervous temperament or are easily excitable or really on the muscle when they work, those horses oftentimes are more susceptible to recurrent exertional rhabdomyolysis, a form of tying up that we think is related to a stress inducing the condition, and they develop muscle disease, we think, because all of a sudden they release an excessive amount of calcium from the storage sites within the muscle cell. It causes the muscles to contract and they don't relax. And then it also incites enzymatic kind of destruction of the muscle, and they develop tying up. So if we've got that history, we've got the right breeds, we've got the right environmental circumstances, and those horses, then we take an approach to manage RER. And then there are horses that we have taken muscle biopsies from and looked at and felt like they had another form of glycogen storage disease, of which we have yet to find the answer, and called that PSSM2. And that's a muscle biopsy diagnosis that we make in some cases where it's an active field of investigation to try to figure out, is there one muscle disease under that category, PSSM2, or are there likely several different things? And it's just a description histologically of this is what the glycogen looked like when we looked at it under the microscope. So it's good to be able to try to make some of those distinctions based on the history, knowing you've got exertional rhabdomyolysis by measuring muscle enzymes, genetic testing where the breed is appropriate, then you can kind of figure out management strategies. The tougher cases to work on now are the ones, particularly warm blood horses, where owners are reporting that the horses just got exercise intolerance, reluctance to move forward, reluctance to collect, but their muscle enzymes are normal. And so trying to tell whether or not this is a muscle disease is really a big challenge, because you don't have a nice blood test that you can rely on that will definitively say, yep, this is actually a muscle disease. And those are really challenging too, because the signs that the horses show could be caused by many things. They could be caused by saddle fit errors that make them cranky and not want to go forward, training issues, ring sour, lameness, any other causes of pain that the horse might have. So those cases really require an extensive investigation by a veterinarian to rule out the most common things first, which are the training issues and the issues with saddle fit and lamenesses. And then when all of those things have been exhausted, one of the diagnostic possibilities is to do a muscle biopsy and try to see if we've got evidence of muscle disease in that muscle biopsy. And in those cases, what we most commonly came up with initially was a diagnosis of type 2 PSSM in warm bloods. And with our subsequent research into that, it seems like there may be a specific underlying disorder that we've now termed a myofibrillar myopathy. And that term came from looking at the samples under the microscope and with electron microscopy and seeing they've got aggregation of proteins that are tasked with trying to keep all of the myofibrils, the contractile proteins, in alignment, and that we've got some disarray of those myofibrillar proteins inside the muscle cells. So we've now got some very special stains that we can do beyond just looking at the glycogen to help try to identify that myofibrillar myopathy. And so for each one of those specific diseases now, we're trying to figure out what's the best training approach and diet approach to handle them separately. One of the most recent investigations we've done is to see whether some of the commercial genetic tests that are offered now for type 2 PSSM and for myofibrillar myopathy correspond to the histological diagnosis of type 2 PSSM or myofibrillar myopathy. So in that study, what we did was to take muscle biopsies that had been submitted to us by and then do all of the special stains that would identify myofibrillar myopathy, the new stains that would pick those up, and do the stains that identify type 2 PSSM. And then we also then, in our own lab, ran the, looked at the genetic mutations that have been reported to and are now offered as the commercial genetic tests. So we looked at 54 warm blood horses that didn't have any abnormalities in their muscle biopsies, 68 warm blood horses that we had diagnosed with PSSM2 or myofibrillar myopathy based on the muscle histopathology. And we also looked at this in Arabians because they develop a form of myofibrillar myopathy, 30 healthy Arabians and 30 that we had diagnosed with PSSM2 or MM by muscle biopsy. And when we did that and we looked at the results of the genetic tests that we ran in our lab, we did not find a correlation between having the genetic mutations and a diagnosis of MFM or PSSM using the method by which those diseases were discovered. And then we also looked at the presence of those genetic variants in 205 horses that had DNA banked in publicly available databases. And we found that it was just very common to find the variants P2, P3, and P4 that are offered as genetic tests for myofibrillar myopathy in the kind of horse population. So we didn't find with our research that we could recommend using genetic testing for PSSM2 or MFM because we didn't have a strong association between the genetic variants and the muscle biopsy diagnosis.
Okay, that's really interesting. We will make sure and put a link to that research in Equine Veterinary Journal in the article that goes with this podcast. So if you're listening to the podcast, go to equimanagement.com under Disease Du Jour and look for this specific podcast, and it'll have a link to that research if you want to read it. And it's open access. Today's Disease Du Jour podcast is brought to you by Merck Animal Health, the makers of prestige vaccines, Banamine, Panacur, Regimate, Protozil, and other trusted equine health solutions. Merck Animal Health works for you and for horses. Learn more about Merck Animal Health's comprehensive portfolio of products, as well as their ongoing investment in our industry, profession, and community at merckanimalhealthusa.com. So let's talk a little bit about, to veterinarians, these tools that you mentioned and when should they use?
Well, so a lot of it depends on the horse owner that you're dealing with. But the genetic testing that are offered, like the 5-panel tests that are offered for quarter horses that have HYPP, glycogen branching enzyme deficiency, which is a problem in foals, a muscle disease in foals, not adults, because it's a fatal disorder, PSSM1, and then a new muscle disease that we've identified called myosin heavy chain myopathy. Those are all offered as a standard muscle panel by many companies now. So they're a nice way to try to sort of screen for underlying susceptibilities. The myosin heavy chain myopathy is a genetic mutation in myosin. And it turns out that genetic mutation in myosin is responsible for immune-mediated myositis in quarter horses. So somehow the mutation causes the immune system after certain environmental triggers to look at the fast-twitch myosin in the muscle and say, you're foreign, I'm going to take you out. And they get all kinds of inflammatory cells in the muscle, and their muscles will atrophy really rapidly. Within 3 days, they can lose, 30, 40% of their muscle mass.
Wow.
Yeah, it's a stunning disease. And it's called a co-dominant disease because one copy of the mutation will provide susceptibility to that disease, but two copies make them even more susceptible and that make it more recurrent and make the atrophy worse. The tricky part with that is understanding why and when horses all of a sudden develop the atrophy. So some horses, we don't know yet what percentage of horses that have that genetic mutation will go on to develop muscle atrophy. That's something that we're interested in doing some surveys to try to understand. And then the other thing that disease does is, the reason we call it a myosin heavy chain myopathy, because it doesn't just show up as immune-mediated myositis. In some horses, it shows up as just severe rhabdomyolysis, so severe tying up not associated with exercise. You'll just find them, it occurs particularly when they're young, but it can occur at any age. They'll come in and they'll just be stiff, sore, moving slowly. And again, it can progress to them lying down and being unable to get up, and can be fatal at times because of that mutation. And exactly why it causes these two different forms of clinical signs, we don't know, but it's this myosin heavy chain myopathy. And it's really a relief to me now that we can just do a genetic test for that and figure out whether or not it's likely. About on average, 10% of quarter horses will have one copy of that mutation, but it's more common in reining horses. Up to 20%, I think it was 18 to 20% of reining horses might have a copy of that. So the next study that we want to do is to start a survey asking people that submit the genetic test to UC Davis, what their horse's history is, and try to figure out how frequently a horse that has the mutation actually goes on to develop clinical signs, and if they know what might have predisposed horses to this. We think, for example, that strep equi, it has a lot of overlap in some similarities between the myosin and the M protein in strep equi. We think that's a potential trigger for this disease. We don't recommend vaccinating for strep equi if a horse is genetically susceptible to the disease. And then sometimes anything that seems to stimulate the immune response. So even other, pneumonia, pigeon fever, that can also potentially be a problem. And also, we don't recommend using vaccines that seem to cause a lot of swelling and muscle damage, because if you expose the immune system to that myosin, it could trigger the condition. So using intranasal vaccines wherever you can, and being cautious not to administer irritating intramuscular injections are kind of our thoughts on that.
Oh, that's very interesting. Make sure and let us know when you're doing that, and we'll help spread the word through Equimanagement to the veterinarians.
Great.
Yeah. Is there any other research that you would like to talk with our veterinary audience about, that either you could use their help or you would just like to pass it along of what Michigan's doing?
We've been doing kind of the approach to doing a lot of our research lately has been to try to investigate the interactions between the genes that are being expressed, the proteins that are being expressed, and the kind of the happening that's going on in the muscle of horses that are susceptible to diseases. And we've worked with Dr. Clara Fenger in Kentucky and found some really exciting findings recently in thoroughbreds with recurrent exertional rhabdomyolysis, kind of confirming that the thoroughbred horses in that race training environment, which tends to be more of a high stress environment, the underlying thing that we see, even when they're not tying up, is that susceptible horses have differences in the genes that they're expressing and the proteins that are expressing that are affecting two main areas. One is the storage of calcium and the release of calcium from the sarcoplasmic reticulum. So what we think happens with RER now is that in a high stress environment, it alters the proteins that release calcium from the storage sites. And so when they get hit with a lot of stress, they're more likely to leak too much calcium out of those muscle cells. And that then the mitochondria try to take up the calcium to prevent it from overloading. And so they have changes in the proteins involved in calcium release, and they have the changes involved in a number of the enzymes involved in the mitochondrial respiratory chain. And interestingly enough, we found when we looked at those horses that were treated with dantrium before exercise, which slows calcium release from the storage sites, those horses did not have those changes in gene expression that we saw in the horses that were not pre-treated before exercise. So it looks like there is a pretty strong component of abnormal calcium release, and that that has a secondary consequence on some of the mitochondrial proteins. So our continuing research is to look at, are there ways we could support the mitochondria? We're looking at called CoQ10 that Kentucky Equine Research produces, and trying to see if that has, what effects that has potentially on mitochondria, working with Dr. Sarah White at Texas A&M University. And we're also looking, funded by the Trotting Association, we're looking at whether we're seeing the same sorts of changes in standardbred racehorses, and whether standardbreds and thoroughbreds have the same underlying basis for RER. And then the other thing that we're working on with Kentucky Equine Research is developing a new product for myofibrillar myopathy. So what we have found with that same sort of integrated approach, looking at gene expression and protein expression with myofibrillar myopathy, is that there appears to be an alteration in the muscle's response to training, where you would normally see activation of genes that will turn on the muscle's ability to make specific proteins in response to training. That system doesn't seem to be working in the warm blood horses appropriately, which might be why we've got changes in the myofibrils. They're not getting strengthened to the extent that they should be with each training bout. And we also are seeing changes that suggest that there's some oxidative stress in the muscle. So we've developed a pellet called MFM pellet that's just become commercially available through Kentucky Equine Research, that has branched chain amino acids and N-acetylcysteine in it to provide the cysteine-based antioxidants that they need. They seem to be breaking those down in a fashion that they shouldn't be, and then to provide branched chain amino acids to stimulate muscle cell development. They need to keep making more sarcomeres and contractile proteins to support those muscle cells. So we're excited about that. Our preliminary results look really nice. And then we're also recommending something very different than what we used to recommend in terms of exercise regimes for horses with myofibrillar myopathy, the warm blood horses. We're recommending that they do an exercise session that's 3 days of work and 2 days off, rather than we used to say exercise horses every day. It seems like they tolerate that exercise schedule much better. So lots of warm up, long and low work with those horses, and then gradually, once their muscles are loosened up, doing some collection work, and then kind of expecting that day 2 is usually the best ride you're going to get. Day 3 is not quite as good and fluid and flowing as day 2, and then you give them a couple of days off, and then they'll come back and work quite well. So we're excited we've got a new approach for that, because we don't find in those horses, we were recommending a low starch, high fat diet, and we didn't find that that was really benefiting them. And so now we're saying don't get the starch too low. They don't probably need a lot of extra fat, and that giving them a little bit of carbohydrate might help that lack of energy and lack of willingness to move forward, as long as you're adding things like the amino acid substitutions and some antioxidant support. So we're trying that coenzyme Q10 on those horses too, to provide extra antioxidant support. So that's kind of the newest. It's a big change from what we used to say, which was low starch, high fat, to no, not anymore. Let's try moderate starch, only fat if the horses need it for weight, which they usually don't. And then adding this amino acid supplement and adding more antioxidant support.
Well, that's great. Is there anything else that you'd like to add to our veterinary audience? I know that you have always got so much going on, but it's wonderful to hear some of this new research and new management techniques that vets can share.
Yeah. The only other thing I would share is we are continuing to see an increase in horses that have vitamin E deficiencies, and chronically over time, when they're deficient in vitamin E, there seems to be two different things that evolve. One is the vitamin E affects the nervous system, and classically you'll get equine motor neuron disease, which most vets are familiar with. But in some horses, what you see initially is just a kind of a decline in performance and a slow and gradual loss of muscle mass. And that seems to be more of a muscle-based issue than a nervous system-based issue. And we have found in those horses that we can make a diagnosis by looking at the mitochondria in the muscle that's just above the head of the tail. That muscle that moves the tail is a slow twitch muscle that has a lot of mitochondria. And when they have vitamin E deficient myopathy or vitamin E responsive myopathy, they will develop changes in the mitochondria that we can see in muscle samples from the sacrocaudalis muscle above the tail. And the nice thing about that is if you catch it early enough, usually those horses will redevelop their muscle mass when they put on vitamin E. But they don't always have low vitamin E at the time that you are evaluating them. So they may have had vitamin E deficiency in the past, but you can't always diagnose that condition based solely on low blood vitamin E. So sometimes if you're wanting to rule that out, we look at muscle biopsies from that particular muscle, the sacrocaudalis muscle, to find that vitamin E responsive myopathy.
Well, thank you very much, Dr. Valberg, for
You're welcome.
being a guest on today's episode of Disease Du Jour. I always learn a lot personally, and I know our veterinarians love to hear what's coming out of your lab.
Great.
And we want to thank our listeners for being with us today for Disease Du Jour, and a special thanks to our 2020 sponsor, Merck Animal Health. Please visit equimanagement.com's latest Disease Du Jour episodes and take our survey so we know how to better serve you with our podcast in 2021. If you have questions or suggestions, you can also send me an email at kbrown@aimmedia.com. Disease Du Jour is a production of the Equine Podcast Network, an entity of the Equine Network.